Hello, I'm Hannah Sachs, Editor-in-Chief of NEJM Evidence, and this is StatStat.
You're hooked watching the Grand Prix when the camera zooms in on a pit stop on the side of the track. The precision and speed are mind-blowing as a crew changes the tires, refuels, and makes other adjustments. As you watch competitors surge ahead while the car is stopped, you realize that drivers shouldn't just stop on a whim. Too many stops and they risk coming in last. Too few and they risk running out of fuel or wearing out their tires. How often should they stop? When? What exactly should each stop entail? Investigators face these same questions when considering interim analyses in clinical trials. Just like in a pit stop, interim analyses allow teams to assess if the trial is on course, monitor participant safety, and decide if adjustments are necessary. But without careful planning, interim analyses can compromise the trial's results. To look more closely at the trade-offs, let's start by discussing 3 reasons to perform an interim analysis. First, to evaluate safety. It is critical to identify potential harms or unexpected adverse events early. Often an independent data and safety monitoring board, or DSMB, reviews accruing data. Say you're conducting a trial comparing a new antiarrhythmic drug with placebo among patients with atrial fibrillation. You plan to recruit 1,000 participants, but an interim analysis performed after 50 patients are enrolled shows that 8 of 25 patients in the intervention group developed complete heart block requiring a pacemaker, compared with 0 in the placebo arm. The DSMB recommends the trial stop immediately given this unexpected harm, and you are glad you learned about it early on. Second, to assess efficacy. Is the treatment showing signs of preventing major adverse cardiovascular events? For instance, say after enrolling the first 500 patients in your trial, an interim analysis shows 10 major adverse cardiovascular events in the intervention group, compared with 50 in the placebo group. This seemingly massive difference prompts the DSMB to recommend early termination of the trial due to overwhelming efficacy. A similar analysis could be used to assess futility rather than efficacy. That is, whether continuing the trial is unlikely to demonstrate a treatment effect, no matter how many patients are enrolled. Third, interim analyses allow investigators to confirm the accuracy of the sample size calculation. For example, your plan to enroll 1,000 participants was based on an overall expected rate of 20 events per year in the trial population. However, your interim analysis revealed an actual event rate of 10 events per year. Recognizing this discrepancy provides an opportunity to recalculate the needed sample size or extend the trial duration to accrue enough outcome events. But some of these benefits of interim analyses come with a cost. First, when a clinical trial is designed and powered for the primary outcome, typically the alpha is set at 0.05. This value represents the probability of making a type 1 error, or false positive, by incorrectly rejecting the null hypothesis that there is no difference between treatment groups. Each interim analysis consumes a piece of the trial's type 1 error budget. Statisticians call this alpha spending. As a result, the final analysis must meet a more stringent significance threshold to account for the multiple testing and to maintain overall alpha control. Second, analyzing data from a trial that was stopped early risks overestimating the treatment effect. Stopping a trial early likely means accruing fewer outcome events than planned, making estimates more fragile, and perhaps by chance, making between-group differences seem larger than they really are. Trials that are stopped early may also have shorter follow-up, missing out on capturing long-term information. To address these issues, interim analyses should use pre-specified criteria for decision-making. In your trial, you originally planned 2 interim analyses, so 3 total looks at the data. 1 after the 50th patient was enrolled, 1 midway through, and 1 at the end. Let's consider 2 approaches to controlling alpha during these analyses. One approach would require a very low p-value for the first interim analysis, a higher p-value for the second, leaving just less than 0.05 for the final analysis. With this approach, interim results would need to be very persuasive and statistically robust for the trial to be stopped early, and it keeps the significance level at the final analysis close to the desired 0.05 threshold. An alternative approach offers more flexibility for stopping early by spreading the alpha equally. A p-value of less than 0.0221 would be required at each of the 3 analyses. This means a higher chance of the trial being stopped early. But if the p-value at the final analysis is, say, 0.04, greater than the 0.0221 cutoff, it would not meet the criteria for statistical significance, a threshold that would have been reached had no interim analysis been performed. The decision about which approach to use depends on the trade-offs investigators are willing to make. So think of interim analyses as the pit stops of clinical trials, scheduled moments to evaluate progress, ensure safety, and make critical decisions. But just like in racing, they require planning, precision, teamwork, and a steady hand on the wheel. When done right, they keep the trial on track and ensure the best outcomes for patients and the scientific process alike.